Welcome to the second part of our digestion lecture. Now we're going to pick up where we left off on our previous lecture, that being the stomach. So just to recap, we finished the stomach talking about its location, its anatomy, its histology, and its function. Now we continue down our tube and we see that we pick up with the small intestine. Now remember, we already introduced that the small intestine is made up of three parts. We will identify those three parts and carry through the same process of looking at its location, structure, histological, anatomical, physiological functions, etc. So let's start. Our small intestine is where we're going to see the majority of digestion and absorption occurring. Now remember that the stomach is also digesting. We see the majority of chemical digestion occurring here with mechanical digestion, the bulk churning. Now we can look at detail of the small intestine. So we start with the first and smallest portion, the duodenum. It's roughly 8 inches long. It is the first and shortest segment of the small intestines. Its job is to receive that partially digested food from the stomach known as chyme. It's also going to play a vital role in chemical digestion of this chyme, which is going to help prepare it for absorption in the further segments. The next portion is the jejunum. Now this is going to be the longest portion of the small intestine. It's going to make up about 2/5 of the small intestine. Its main function is going to be absorption, so the majority of absorption occurs right here in the jejunum. The specific nutrients that it's going to be absorbing are going to be sugars, fatty acids, and amino acids. The last section is the ileum. Now, the function of the ileum is to absorb nutrients that did not get absorbed by the jejunum. Now, these nutrients are going to be super important. We're talking about vitamin B12, and we're also reabsorbing bile acids that we're going to reuse later. We can elaborate on the functions of the small intestine. We see that we get segmentations, which is going to mix the chyme with the digestive juices and bring food into contact with mucosa for absorption. Now, if we go back to AP1, we talk about that anatomy. Remember, we did the small intestine for simple columnar epithelium, and if you think to the modifications we saw on this epithelium, we saw goblet cells for mucus production, we saw Brunner's glands or duodenal glands. And we also saw microvilli. So if you go back and think back to those finger-like projections on top of finger-like projections on top of finger-like projections. So what's our goal? Maximizing surface area. Why are we doing that? To maximize absorption. So peristalsis is going to be the mechanism, the smooth muscle contractions we're going to use to help propel that chyme mixture through the small intestine. And here, mixing with enzymes from the pancreas, from the liver, and subsequently the gallbladder, we're going to complete digestion of carbohydrates, proteins, lipids. And we're also going to begin and finish digestion of nucleic acid. Remember, one of the functions of the pancreas was to secrete nucleases, which is going to break apart those nucleic acids. We're going to absorb about 90% of the nutrients and water that pass through the digestive system. Now, if we look at the internal anatomy of the small intestine, we already alluded to those finger-like projections on top of finger-like projections on top of finger-like projections. So we have to realize that the small intestine changes. The tube remains the same, but the internal anatomy changes. And the reason why is this. Think about our goal. Our aim is to slow things down, to optimize and give digestion enough time to occur, and also to give us enough surface area and enough time to absorb all those nutrients. So the internal anatomy, those first finger-like projections or plicae, is going to be arranged in a circular arrangement. So we're going to call them plicae circulares. These are circular folds. So those first finger-like projections, those first plicae, will then give rise to those villi, the next finger-like projection, which give rise to those microvilli. Again, maximizing surface area. Why circular folds? Think of it like a corkscrew. The chyme mixture is going to corkscrew its way down that small intestine. So you say, why? Why not make it a straight tube? Well, if you've ever been to any store and you've seen those gumball machines that you drop the money in and then the gumball corkscrews its way down. What this does is essentially slows the time that it takes to get the gumball from point A to point B. Instead, if it was a straight tube, the gumball would drop straight down. It would take a microsecond and you'd have that gumball in your hand. So what our body does is the exact same thing. It is slowing down this chyme mixture, exposing it to all those villi and those microvilli, maximizing digestion and maximizing, again, absorption. That brings us to the histology of the small intestine. So we can start at its most macro level, where we see just roughly the location of the entire small intestine. You see it's surrounded by the colon and it is situated in the abdominal cavity right here. We can then take a nice little cut and look at those plicae circularis, and from here we go to some histological components. So we can see we have our same layers, right? We have our mucosa, we have our submucosa, our 2 layers of muscularis, and again, because we're surrounded by a mesentery, a serous membrane, we have a serosa. Now we can look at all the various types of cells, and we're going to stay pretty general here because in our next slide we're going to get a little more detailed. But we have 4 different types of cells in this mucosal area, and we'll talk about those in a little bit. But notice the arrangement, and they are differentiated by color. So we see some lighter ones here, some darker ones here. We have some blue ones and some yellow ones. So we'll talk about all these absorptive cells, secretory cells, etc. So we'll talk about these in a little bit. But a couple more things I want you to point out. We have goblet cells, okay, so we are producing mucus here. We have a lamina propria, same arrangement that we have holding up this mucosa and giving it some support. We have our enteroendocrine cells that we're going to talk about in a little bit, so that's our type 1. We're going to have Panna cells 2. We're going to have a lymphatic nodule, so yes, we have lymphatic here. And I'm going to go back up because I skipped this one on purpose. We go to a lymphatic capillary. Let's talk about this lacteal for a little bit. So this lacteal's job is going to be to absorb dietary fat. And it's going to pick that up in the form of chylomicron. And again, we'll talk about this in a little bit. To end our mucosa, we have our muscularis mucosae. That brings us to our submucosa. And in there, we're going to find our arterioles, our venules, our lymphatic vessel that give rise to that lacteal that moves into the villi. We have our 2 muscle layers, our circular layer or longitudinal layer with that myenteric plexus right in between. So we hit off that muscularis, and then we end with our serosa. Intestinal juices and brush border enzymes. What exactly do these do? So intestinal juices provide a vehicle for absorption of substances from that chyme mixture as they come into contact with the villi. Those brush border enzymes, they're going to be found on the surface of the microvilli of the absorptive cells. And their job, again, is going to be to break down food products. So again, continuing some chemical digestion as well as absorption. Let's take a look at the histology of the small intestine. We're going to go into a little further detail with our cells and what exactly they do. The first cell we're going to start with are our microvilli. And again, if you remember from AP1, we talked about this being a modification for absorption. So these are our absorptive cells. Their goal is to digest and absorb nutrients. Our next modification that we saw in our epithelium was our goblet cell. And like we talked about previously, this should be a repeat for most of us. This is going to secrete mucus. The next cell we're going to deal with is an enteroendocrine cell. And as its name implies, it's a cell located within a structure, and it's going to have an endocrine function. So what's it going to do? It's going to secrete hormones. It's going to secrete secretin. And secretin's job is going to be to regulate gastric acid secretions as well as pH levels in the duodenum. Remember, the small intestine is broken up into our 3 parts. The first 8 inches or so is the duodenum. So we're talking about the first few inches of the small intestine that this hormone is taking action. The next hormone we look at is cholecystokinin. And if we break this word apart, we find 3 distinct Latin derivatives. Chole, meaning bile, cysta, meaning sac, and kinin meaning movement. So if you put this together, this hormone is dealing with the movement of the bile sac. So now if we go back and think about our anatomy of the liver and the gallbladder, remember that the gallbladder is the sac that is storing bile. So now we can put this together and we say that cholecystokinin's main function is going to be to stimulate the release of bile into the lumen of the intestine, of the small intestine particularly. And then it's going to aid in the secretion of pancreatic enzymes. Now the third and final hormone that's going to be secreted from these enteroendocrine cells is going to be GIP. Now GIP is also called glucose-dependent insulinotropic polypeptide. Now, levels of GIP rise immediately after the ingestion of nutrients. What this does is it leads to a modest inhibitory effect on gastric acid secretion. And it's also going to inhibit gastrointestinal motility. So to summate GIP, its name can also be called gastric inhibitory polypeptide if this helps you to understand that this is an inhibiting hormone other than a stimulating hormone that we have talked about before. And we did say that its action, although weak, modest, is to inhibit gastric acid secretion and motility of the gastrointestinal tract. Its main role is going to be to discuss and talk with the pancreas to stimulate insulin secretion. And finally, the last cell type we're going to look at are Paneth cells. These guys are very similar to neutrophils in which they're going to provide our small intestine defense against microbes. They do this by secreting lysozymes, which is going to allow this cell to be capable of phagocytosis. So finding any pathogenic structures, bacteria, virus, etc., these guys can come in and gobble them up. Now we can look at the histology of the small intestine. Now, we're going to look at two different portions here. We're going to look at the duodenum and the ileum. And the reason being is because in the submucosa, and partially extending into the mucosa, we're going to find glands, different glands in the duodenum than we see in the ileum. So let's start right on top right here. So we start with our duodenum and we again break down our four basic regions: mucosa, submucosa, muscularis, and serosa. Now, key here, in the duodenum, we find submucosal glands. These are also known as Brunner's glands. Brunner's glands. Now, the point of Brunner's glands is going to be to secrete an alkaline-based mucus. Now, think about that. An alkaline-based mucus. This is a neutralizing mucus. Now, what its job is going to be is to protect the lining of the small intestine, of the duodenum. So look, remember, the acidity of that chyme mixture that's leaving the small intestine is relatively low. It's really acidic. Now, if we don't have secreting cells or glands that are there in place to protect the small intestine, that acidic chyme would digest the small intestine. So we have these Brunner's glands whose job is going to be to secrete this basic fluid, again, neutralizing that chyme mixture coming out of the small intestine. As we travel down past the jejunum into the ileum, we look at the histology as it changes. So again, four layers are going to stay the same. The mucosa, the submucosa, the muscularis, and the serosa are going to go unchanged. The big change is in the submucosa, these lymphatic nodules. Now, these lymphatic nodules, we're going to name them. So here's our lymphatic nodules, right? These big, large purple masses, these purple balls. What are they for? They are called Peyer's patches. And remember, we introduced Peyer's patches when we did our lymphatic lecture, but we know that they are aggregated lymphoid nodules, and their job is going to be to surveilling. They're kind of like our X-ray center of our small intestines. If you go back to our lymphatic lecture where we say that our lymph nodes and lymphatic structures are kind of like the security center in an airport that is surveilling, checking, monitoring, adjusting, protecting the travel. Well, our lymphatic system is doing the same thing. It's protecting that fluid that's being distributed back into our blood by screening, killing, phagocytizing anything that shouldn't be there. Well, this is doing the same thing. So Peyer's patches are monitoring that intestinal bacteria population. Remember, we have symbiotic bacteria inside our small intestines. And it's monitoring this. It's also monitoring the bacteria in what you're eating. And it's preventing the growth of pathogenic bacteria in the small intestines. Now we say, well, what's a pathogenic bacteria? Again, remember, pathogen is any substance or protozoan, bacteria, virus, anything that wants to do harm to your body. And we can talk about Salmonella as a pathogenic bacteria. E. coli is a pathogenic bacteria. So this is what these Peyer's patches or lymphatic nodules are surveilling or checking for. We've spent a lot of time so far, and we're going to look at it some more, the chemical digestion in the small intestines. Now let's look at the mechanical digestion. How do we move this chyme liquid through the small intestine? Now, the method that predominates here is segmentation. Now remember, unlike peristalsis, which predominates in the esophagus, this segmentation is going to be localized contractions, which is going to predominate in the small intestine. Now, these localized contractions are going to utilize those circular muscles in the muscularis layer to help propel this chyme mixture down, back and forth through those plicae circularis. Finally, peristalsis is going to work in those wave-like propulsive contractions. Now, typically in the small intestine, they're going to appear as irregular intervals and travel for, well, varying distances. Some of these peristaltic wave contractions can travel a few inches, other can move a few feet. Their ultimate job is going to be to expose food to the intestinal wall for absorption and to assist in moving it forward. Chemical digestion in the small intestine. So we're going to examine exactly what is being broken down and the substrates of this process in our subsequent slide. So let's first introduce it. So what we are digesting, or at least continuing digestion in the small intestine, will be 1, carbohydrates, 2, proteins, 3, lipids, 4, nucleic acids. Now, this list should bring us back to the pancreas and those digestive enzymes secreting. We're going to see that pancreas assisting us with digestion and further breaking down some of these complex nutrients. Table 24.4 summarizes the digestive activities in the pancreas, liver, gallbladder, and the small intestine. The nice thing about this slide is it tells us and breaks down these cells, the organs, the layers, and what structures are responsible for specific activities. So for example, we look at the pancreas. We see that it delivers pancreatic juice into the duodenum via those pancreatic ducts to assist absorption. Refer back to 24.5, or at least we're going to look at 24.5 in a little bit to see those pancreatic enzymes and what exactly they do. The liver, its job, producing bile salts, which is necessary for emulsification, remember, breakdown of lipids as well as absorption. The gallbladder is going to be our storage tank, storing concentrates and delivering bile into the duodenum via that common bile duct. That brings us to our small intestine. And in our small intestine, we see a host of regions, at least words we have discussed already, just nice and neat package to break it down. The mucosa and the submucosa, we see the intestinal glands. They're secreting intestinal juice to assist in absorption. Absorptive cells doing exactly what their name implies, digesting and absorbing nutrients. Goblet cells, refer back to AP1, modifications of epithelium, where we're secreting mucus. Our enteroendocrine cells, we have secretin, cholecystokinin, and GIP. All these are going to be secreted to monitor and control what's going on in the stomach and the small intestine. Panneth cells secreting lysozymes. So we're going to look at these in a little bit. So those Panneth cells are secreting lysozymes. These are bactericidal enzymes that are destroying bad bacteria and phagocytizing anything that does not belong there. Brunner's glands, they're secreting an alkaline fluid to help buffer the stomach acids and mucus that's protecting and lubricating. Our plicae circularis, so we start here, plicae circularis, folds of the mucosa and submucosa that are increasing surface area for digestion and absorption. We have our finger-like projections called villi, maximizing surface area, and then our finger-like projections on top of that, microvilli, so microscopic membrane cover projections of absorptive epithelial cells that contain brush border enzymes, increased surface area for digestion and absorption. And then we have our muscularis layer, our segmentation, types of peristalsis, alternating contractions of circular smooth muscle fibers that produce, there's our word, segmentation. Remember, the majority is going to occur through segmentation and resegmentation of sections of the small intestine. This is going to help mix chyme with digestive juices and brings food into contact with the mucosa for absorption. Migrating motility complex, MMC. This is going to be our peristaltic, waves of contraction and relaxation of circular and longitudinal muscles, which are passing the length of the small intestine, again, moving chyme towards the ileocecal sphincter. When we get to the large intestine, this is a new word for us, ileocecal sphincter. We'll find this little sphincter, ring of muscles, that is connecting or allowing for movement of that chyme from the small intestine, specifically the ileum, into the cecum. Table 24.5. This table is going to summarize our digestive enzymes. Now, while there's a lot more information in here than we are required to know, we are going to go and spend some time talking about the enzymes, what activates them, sources, substrates, and products. Again, some of these products we could keep some general knowledge. We're not going to go too crazy with specific names of disaccharides and trisaccharides, things like that. So let's start with saliva. We know that we are secreting two enzymes, one salivary amylase. From the salivary glands, we're breaking down starches. So again, kicking off polysaccharide digestion. The product is going to be, we take it one step down. So polysaccharide or trisaccharide into maltose, which is a disaccharide. Or if we take other types of polysaccharides, we could break it down into maltotriose, which is a trisaccharide, and alpha dextrins. Next is lingual lipase. Remember, we're dealing with fats and diglycerides or triglycerides. So we're starting with lingual glands or the sublingual glands of the tongue. Our substrate, what we're breaking down, is triglycerides and other fats. And our product are going to be fatty acids. And again, one step down from triglycerides into a diglyceride. That brings us to our stomach, our gastric juices. Pepsin, remember that pepsin's activated from pepsinogen. So we're taking hydrochloric acid and pepsinogen to give us pepsin. Those chief cells are responsible for that. We're breaking down proteins into peptides. That brings us to gastric lipase. So again, we're seeing more fat from the chief cells of the stomach. So chief cells, we're breaking down triglycerides, fats, and oils. And our product is going to be fatty acids and monoglycerides. And that brings us to the pancreas. Our pancreatic juices, we start with pancreatic amylase. This is secreted from those pancreatic acinar cells. What are we breaking down? It's similar to salivary amylase. So we're breaking down starches, polysaccharides, breaking them again into disaccharides and trisaccharides. Trypsin is going to be activated by trypsinogen, and this is going to be by enterokinase. We're going to see pancreatic acinar cells are going to be secreting this trypsin. That's going to help the break down proteins, the product, peptides. Next is chymotrypsin. This is going to be activated from chymotrypsinogen by trypsin. The pancreatic acinar cells are secreting this as well. We're breaking down proteins into peptides. So you see the importance of protein degradation here. Next is elastase. This is going to be one of our final protein or proteolytic enzymes. We deal with elastase being activated by proelastase, and again, this is by trypsin. The source is our pancreas, protein is our substrate, and the byproduct or the end product is going to be peptides. Carboxypeptidase, this is going to be activated from procarboxypeptidase from, again, trypsin. We have the pancreas, the acinar cells of the pancreas doing all the work. Amino acids at the carboxyl end of peptides is our substrate, followed by amino acids and peptides, again, for our product. Pancreatic lipase, so we're seeing the acinar cells of the pancreas secreting lipase. to break down triglycerides, other fats and oils that have been emulsified by bile salts into our product being fatty acids, and look at our result from polyglycerides all the way down to our monoglyceride. We have nucleases breaking down ribonucleases and deoxyribonucleases. Again, pancreatic acid in our cells, we're breaking down ribonucleic acid and deoxyribonucleic acid, so RNA and DNA, with our end result being nucleotides. Now to finish table 24.5, we're going to look at the enzymes that are secreted from the brush border in the microvilli in that plasma membrane. We start with alpha dextrinase. So again, the source is going to be the small intestine, specifically those microvilli. We're going to attack our alpha dextrins, and the byproduct is a simple sugar, a monosaccharide. Now we can look at maltase. Again, its source is those microvilli in the small intestine. The substrate, what it's going to be digesting, is maltose, and it's going to be breaking it into a simple sugar, glucose. Sucrase is going to be, again, source is all the same, small intestine. We are breaking down sucrose into glucose and fructose. Lactase is, again, same place. We are breaking up lactose. You see the similarity here: sucrase, sucrose, maltase, maltose, lactose, breaking it into glucose and galactose. Now the interesting one here is enterokinase. We talked about this before when we were activating trypsinogen and forming trypsin. So that enterokinase is going to be super important. Of our peptidases, we have aminopeptidase and dipeptidase. These guys are breaking apart dipeptides, amino acids, and amino acids at the amino end of the peptides, breaking them into amino acids and peptides. Lastly, nucleosidase and phosphatases. These guys are going to be breaking apart nucleotides into their nitrogenous bases, pentoses, and phosphates. Absorption in the small intestine. So exactly how are all these sugars, these amino acids, these peptides, these fatty acids making their way into the cells? So we first look at glucose and galactose. What's happening here? We're going to take glucose and galactose and we're going to use secondary active transport, so ATP derived, to bring it in with sodium into the cell. We use fructose here. Fructose is going to use facilitated diffusion. It's going to make its way into the cell. And then these monosaccharides are going to utilize facilitated diffusion to get into the blood capillary of the villus. From which point, look, we are taking all of these substances. We are using a nutrient-rich blood via the hepatic portal vein, bringing it to the liver to be utilized. Amino acids are dipeptides and tripeptides. For amino acids, we're using active transport or secondary active transport with sodium. We're using secondary active transport with hydrogen for our dipeptides and tripeptides. So we take these amino acids, we're going to diffuse them into the blood capillary, to the hepatic portal vein, to the liver. All our small short-chain fatty acids are going to go via simple diffusion in conjunction with our large short-chain and long chain fatty acids and our monoglycerides, all simple diffusion. The small short-chain fatty acids will diffuse directly into the blood, to the hepatic portal vein, into the liver. Our simple diffusion of this group here goes via and gets brought into as triglycerides. These triglycerides are going to be brought into the blood as chylomicron. It is going to move its way get absorbed through the lacteal, so those lymphatic vessels inside those villi that we pointed to before. So into the lacteal, into the lymph, carried through the thoracic duct, and then brought into the internal jugular. Now, just as a side note, if we look at triglyceride, we look at these levels in blood, can tell us a whole host of health factors, saying if our diet is too fatty, too rich in fats, fast foods, preservatives, etc. These levels will be elevated and can be adjusted because they do increase the chance of cardiac disease or coronary arterial disease. Movement of nutrients. We can put everything we looked at on that chart previous slide into an actual picture. So we start with the villus, so we're really zooming into this finger-like projection. We see our blood capillary, we see our lacteal, and our arterial, so these are our macro structures. Now we can start looking at a nutrient level. Here's that chylomicron being taken up from the blood into that lymphatic vessel, into that lacteal, which point it's going to carry its way through our system and dump back in to that subclavian, left subclavian vein. And then we see our amino acids, our monosaccharides, and our small short chain fatty acids moving into venous circulation. Now again, look at the color. Remember we said for portal circulation, that portal vein is nutrient rich but oxygen poor. That's why we denote it as being that purple color instead of blue. So this gives us a good representation of all the reabsorption of those nutrients into the capillary, into that peritubular capillary, at which point into the venule it will go traveling to the liver. Fluid balance. We don't realize how much fluid is really being absorbed through our digestive tract based on what's being ingested as well as being secreted. So let's start at the top and work our way to the bottom, and we'll see and put a percentage and number to exactly how much is being ingested versus absorbed and released or excreted in our feces. So we start right here in our mouth. Our salivary glands. Our salivary glands produce about 1 liter of saliva a day. Ingestion, we ingest about 2.3 liters of liquid. Our gastric juice, our stomach produces about 2 liters of gastric juice a day. The liver and gallbladder are secreting about 1 liter of bile a day. The pancreas is contributing another 2 liters of pancreatic juices. The intestines are going to be secreting about 1 liter worth of enzymes as well, which gives us a total ingested and secreted volume of about 9.3 liters. Now, the small intestine, remember we said, does the majority of our absorption. We are taking about 8.3 liters through the small intestine. The large intestine is going to take up about 0.9 liters. We need to be precise here because we don't want to absorb all of it. So we get a total absorbed volume of about 9.2 liters, which if we do the math, 9.3 minus 9.2, that gives us 0.1 liters or 100 milliliters of fluid that's excreted into our feces. Now, again, this number is dynamic. This is in a perfect real world scenario, but again, not always is the system perfect. There's bacterial infection, viruses that can contribute to the blocking of the absorption of a lot of this fluid here and here, which can lead to conditions known as diarrhea, or where we are absorbing way too much fluid, and it can lead to conditions known as constipation. As we leave the small intestine, the next player in this series of events is the colon, or the large intestine. So we're going to pick up right here. So we see the ileum right here, and then it's going to travel and form a valve. This ileocecal valve or sphincter is what leads into our cecum. Now, a couple things to point out before we work our way in and around the structures and anatomy of this tube. There's a couple things I want to point out. Number one, hanging off the back of this cecum is our appendix. Remember, this is a vestigial structure, variform appendix. It has some lymphatic structure, otherwise serves no purpose. Our naming of our colon, we have the ascending, means going up, so movement of this fecal matter will move up. We have the transverse colon, going to the transverse, across, and then our descending colon. We hit our curvature. This is going to be our sigmoid colon. From there, we hit our rectum, our anal canal, and our anus. Now, we're going to erase some of this mess so that I can point out some more structure. So bear with me while I erase all the lines I drew. I'm going to leave the directional lines up so that we can still refer back to those. Now, again, material, that chyme mixture comes into through the ileum, in through that ileocecal sphincter, now into the cecum. As we move up, notice the arrangement of the large intestine. There are no plicae circularis. There are no circular folds. There are no villi. We do retain the microvilli, though, because we are still absorbing here. Remember, 0.9 liters of fluid in that water fluid balance is being absorbed here. But notice the arrangement. Bulges. Bulges. These bulges are called haustras. Each bulge is going to serve a function of being compression and then mass peristaltic movement. So moving, compress, compact, and move. We use this band of muscle here. This band of muscle, we see it moving around and down to help with this wave-like motion, this wave-like peristaltic, and it is called the tinea coli. We see it right here. Now, a couple turns. We have a right turn here, a left turn here. This is called the right colic flexure versus the left colic flexure. We're going to call it something different. Colic is more academic. We're going to use a more clinical term because I like where it tells us what organ is lying close to this turn. So we have our right or hepatic flexure. See that word hepatic because we're right by the liver. And our left splenic flexure because this is where the spleen is going to be lying. Then we have our sigmoid flexure. So this is the turn. So sigmoid flexure. And what this is going to give us, so sorry about that, what this is going to give us is to our sigmoid colon. Now that brings us to our rectum. Notice that our rectum zigzags its way through here, forming our anal canal. In here, we're going to have two sphincters, an internal anal sphincter and an external anal sphincter. And this is going to work the same way that our urinary system works. The internal anal sphincter is going to be smooth muscle. This muscle is going to spasm without your control, which leads us to our external anal sphincter. This is skeletal muscle. We see this here. This is going to allow you to be able to control the opening and closing of that anal canal, that anus. So essentially what you can do here, this anal column fills with feces. And as those feces fill, we get this reflex, a distension reflex that tells us, hey, it's time to go to the bathroom. The smooth muscle is going to be contracting and then telling you that you have this urge to eliminate that waste, but you're able to hold that opening in your anus closed because of that external anal sphincter. Once you find yourself at a toilet, you can release that muscle or allow that muscle to relax to allow your feces to flow out into your toilet. That brings us to the functions of the large intestine. So first and foremost, we see this haustral churning. So remember, those bulges are called haustra. So in there, they're compressing, compacting, churning. We get peristalsis and mass peristalsis waves driving the contents of the colon into the rectum. So again, compress, compact, move. We're going to have a symbiotic relationship with bacteria in the colon, which is going to convert proteins into amino acids. So some of these amino acids are very important for us. We're going to break down those amino acids and produce vitamins, specifically vitamin B, or B vitamins. We're going to absorb some water here, and again, we talked about diarrhea versus constipation, and that's regulated based on the water reabsorption. We're going to absorb ions and those vitamins that were produced with that symbiotic bacterial development. We're going to form feces and finally remove and release that waste via defecation. Histology of the colon. So notice that it looks starkly different from our small intestine, which had plicae circularis and villi, but notice that we retain these absorptive cells, these microvilli, that's going to allow us to absorb. So we're going to start macro and work our way in. We start with our four layers, our mucosa, our submucosa, our muscularis, and our serosa. We see that we have openings to this intestinal gland. that leads to these intestinal, almost like pits. So what happens? We have our absorptive cells that are absorbing. We have a massive amount. When we put this under a microscope, and you will do this in lab, you will see that there are tons and tons of goblet cells here. They're going to be lining this surface with mucus to facilitate movement, acting almost like oil on a slip and slide, facilitating these feces to move without being too abrasive. We are going to have some intestinal glands that we'll look at in a bit. We have our lamina propria, so again, what's supporting the lymphatics, the blood vessels, and through the mucosa. We do have some lymphatic nodules here that's surveilling as well. That brings us to our muscularis. Our submucosa. So below this muscularis mucosae, so this band of muscle, right here, we have our submucosa. And again, you see it is connective tissue that is supporting blood vessels, lymphatics, arteries, and veins, arterioles and venules. And then that brings us to our muscularis. Two layers of muscle, a circular layer, our myenteric plexus again, so this is ANS innervation, as well as our longitudinal layer, followed by our outer serosal layer. We could look at the glands and cell types of the colon. So we start with these openings. The openings in the intestinal glands form crypts. These crypts are called crypts of Lieberkühn. In these crypts, we find our intestinal glands. These are going to be secreting mucus. In conjunction with our goblet cells, they're going to secrete more mucus. We found a lymphatic nodule. And again, our absorptive cells. Notice the microvilli. So we have our microvilli that is aiding and causing or allowing these cells to actively absorb. When we look at the histology of these structures, we see again the opening to the intestinal gland. We see our absorptive cell with our microvilli on top. We have a ton of goblet cells. Okay, we're making a ton of mucus here. And again, our intestinal glands that are also allowing for mucus production to occur, while also allowing new cells to be regenerated here because a lot of these cells are abraded with the passing of the feces. Mechanical digestion in the colon starts with haustral churning. So distension reaches a certain point and the walls of the haustra contract to squeeze the contents onward. So if you think of it like a trash compactor that is compressing, squeezing, pushing, and then moving. We rely on peristalsis, these propulsive contractions, to help move and aid in the movement through the haustras, as well as mass peristalsis. So this is a strong peristaltic wave that begins in the transverse colon and then it's quickly going to drive the contents of the colon into the rectum. Ultimately, there is some chemical digestion going on in the colon. The last stages of digestion occur here through bacterial action. So we said before that we have this symbiotic bacteria that is residing in our gut and our colon. These bacteria are breaking down substances that our body was unable to. As a result, we have the synthesis through bacterial action of vitamins, specifically our B vitamins. Thanks to those absorptive cells that line the large intestine, we are actively engaging in absorption and feces formation. So the colon, the large intestine, will be absorbing water, electrolytes, and those vitamins through that symbiotic action with the bacteria. The feces that is formed is going to contain water. It's going to contain inorganic salts, sloughed off epithelial cells. Remember, this food started at the mouth and went through all these other organs. Some of it is very abrasive, at which point it will scratch, abrade, and move some of this epithelium through this tube all the way down through into the colon, as well as the epithelium of the colon. We're going to get bacteria released here, products of bacterial decomposition, and undigested portions of food. Usually, typically things that are cellular based, like salad, corn. If you think of it as when you eat corn, the way it comes out is exactly how it comes in. We do not derive much energy from here. We cannot break through that cell wall. Lastly, we get to the process of removing this feces, all the work that the digestive system did to absorb those nutrients and that fluid and those water and maintain electrolyte balance. We now have to get rid of the waste. We get rid of it through a reflex. The reflex is called the defecation reflex. What happens? Those rectal walls distend and stretch receptors send sensory nerve impulses to the sacral spinal cord, basically saying that our anal canals are full, our rectum's full, it is time to remove the waste in this tube. Motor impulses travel back to the descending colon, the sigmoid colon, the rectum, and the anus. The longitudinal rectal muscles contract and the internal anal sphincter opens. Notice we are in the internal anal sphincter. We have not moved to the skeletal muscle of the external, so the internal anal sphincter opens first. And then when you get to a toilet, you will then control the external anal sphincter and release those contents into your toilet. Table 24.6 summarizes the digestive activities that occur within the large intestine. It goes through the lumen, the activity being bacterial action, and our bacterial activity, and the functions that are occurring, breaking down undigested carbs, proteins, amino acids into products that can be expelled in feces or absorbed and detoxified by the liver. We are synthesizing certain B vitamins and vitamin K. In the mucosa, we are secreting mucus and we are actively engaging in absorption. So the mucus, again, we said lubricates the colon, protects that mucosa. Water absorption solidifies feces and contributes to body water balance. Solutes are absorbed, and this includes ions and some vitamins. Our muscularis, so muscle layer, we are haustral churning, moving contents from haustrum to haustrum. Compress, compact, and move. Peristalsis, move contents along the length of the colon by contraction of the circular and longitudinal muscles. And then that mass peristaltic contraction forcing those contents from the transverse colon into the sigmoid colon and the rectum. And then ultimately our defecation reflex, which eliminates the feces by contraction in the sigmoid colon and the rectum out through the anus. Table 24.7 summarizes the organs of the digestive system and their function. We're not going to go through all of these as this is a repeat, but this is a great study tool for you to use when making note cards and summarizing everything that we've talked about in this previous lecture. Now that we have talked about the entire digestive tract, the anatomy and the physiology, we can now put this process to phases. The first phase is the cephalic phase. So we really say, where does digestion begin? A lot of people say in the mouth, when in actuality, digestion begins with the visual, olfactory, and even auditory cues that we hear, see, or think about food. If you smell a hot apple pie, this will really kick off some parts of our digestion. But then we put food in our mouth, and that whole process of mastication and deglutition gives us the stimulation of gastric secretions as well as motility down into our further parts of or portions of this tube. Next is the gastric phase. This gives us our neural and hormonal mechanisms that stimulate the release of gastric juices from the stomach as well as and leading up to our intestinal phase, which is again neural and hormonal mechanisms that will give rise to secretions from the intestines and the pancreas and the liver and gallbladder, all aiding in the final phases of digestion. Table 24.8 deals with major hormones that are going to control digestion. So again, I like this table because it goes through gastrin, secretin, and cholecystokinin. We've discussed these, but it puts it in a nice little neat package to tell you what the major effect is as well as the minor effect. What I want you to focus on is the major effect here. Don't worry too much about the minor effect, but review it because it should make sense as to what its function is. Our final focus on homeostasis looks at the contributions of the digestive system for all body systems. So ultimately, the digestive system breaks down dietary nutrients into forms that can be absorbed and used by body cells for production and producing ATP and building body tissues. We are absorbing water, minerals, vitamins that are needed for growth and function of our body tissues. Ultimately, we are eliminating waste from the body tissues in feces, and we can see how each of these systems contribute to the digestive system in doing and completing its ultimate goal.